Now I think it's in a full screen.
Yes.
Yeah, good. Good morning, everyone, and a special thanks to all the organizing committee for giving me this opportunity to present my work in this international webinar. So today I would like to present my research work on use of dynamic combinatorial chemistry in drug discovery. And I will give one example, how we find some compounds which can tackle the replication of coronavirus. And I think everyone knows now what coronavirus is, but I will go in details in my next slide. So just to give an overview, what I will cover in my presentation is my short personal journey. I think Madam explained it quite well. I will give you a little bit of more perspective on this field. Then I will discuss about research in European Union, especially about the budgets and which fields they are covering. Then I will give you introduction of drug discovery process and timeline, and the technique which I use, which is called target-directed dynamic combinatorial chemistry. Then I will jump into the results where we find some inhibitors of SARS Co2, which is a coronavirus, using this technique. Finally, the conclusion outlook, and the last, I will show you which are the different research opportunities abroad for young and enthusiastic researcher from India. And finally, the acknowledgements. So, starting my journey, I was born in Untitled. I did my PhD, I did my bachelor from Jagdamba Mahavidyalaya, and then I pursued my MSc in Shivaji Science College, Amravati University. After my master's, I went to Pune, and there I joined National Chemical Laboratory as a project assistant, where I worked for 3 years on different research projects, international and industrial and governmental research projects. And there I got an opportunity to know what are the different opportunities abroad for pursuing the PhD. And during that, I was able to apply for several grants, and one of the grant application was allowed or awarded to me, which was Galileo Galilei PhD fellowship at University of Pisa, where I work on asymmetric catalysis. And after completing my PhD, I work in the same institute as an internal postdoc, and mainly on a nanoparticle synthesis. And then I moved to Milan, which is a northern part of Italy, where I continue my postdoc, and there I work on development of nanoparticle for catalysis purposes. Then after this experience in organocatalysis and metal catalysis, I wanted to pursue further in organometallic chemistry. And there I found a really nice group at University of Groningen, which also hosts a recent Nobel laureate, Ben Feringa. So there I pursued my second postdoc, and I work on development of new reaction methodologies, where we published several research articles in highly ranked journals. Then after all this research work on chemistry, I always thought about what are their applications, like what are a real life application of them. And that brings me to my next application, which is a Marie Curie Fellowship, which is a highly prestigious fellowship in European Union and highly competitive. So where I wrote a proposal to show that how this methodologies, new methodologies which I developed, can be applied in a drug design and development projects. So that I got a Marie Curie Fellowship in Germany, and in Germany I worked in Helmholtz Institute for Pharmaceutical Research, Saarland. This is situated on the border of France and Germany, and this is a really nice picture of my institute where I work, which is situated at the foothills of this nice forest. We have 3 floors. Each floor have a different expertise. So on the third floor, we have a natural product isolation and their use as drugs. On the second floor where I work, we are mainly medicinal chemists. We work on synthesis, development of new drugs, finding the new drug candidates and develop them towards the market or the preclinical studies. On the first floor, we have a drug delivery team. Here, whatever drugs we prepare, they are tested how to deliver them to different animals. So before going to the science, I would like to show how European Union funds research. So if you look at these balls, they are like the countries. So D is Germany, this is UK, this is Italy. This shows what are the average research and development investment from 2004 to 2009 and the GDP growth. So as you can see that the higher the R&D investment, better their GDP growth. So this is one of the things that most advanced countries, they tend to invest more and more money in research and development, and this reflects in their GDP growth. Unfortunately, in last decade, the share of European Union towards the R&D expenditure is falling down, and the China, Europe, and the United States are taking it over. So this is a good trend for the rest of the country, but for European Union, it was a kind of setback. So also, if you see the patent filed from EU, it was kind of now decreasing, and it was increasing from the United States and China. So European Union at the time thought, okay, this is not a good situation, and we should invest more money into the research. So although the patent and the funding was really going down, but the quality of the research was highest. So these are the different countries in European Union, and as you can see, they almost all of them reach their R&D target. R&D target is like when you write a project, you set some targets, right? And that targets are matched by these countries, which is remarkable, especially for the Finland, Sweden, Denmark, Austria, Germany. These are the countries where the most of the research and innovation is carried out. So same thing, the performance of these different countries, as you can see from the right-hand side of this bar chart, there is Sweden, Denmark, Finland, Netherlands, United Kingdom, which is in England, and these kind of countries, and also Germany, they are the innovation leaders. So most of the innovation in European Union are kind of driven by these countries. All the other countries are not bad. They are also above 50%, but you can see these are the leaders. So the Horizon 2020 was the European Union research objective. What they thought at the time that, okay, to boost this kind of research, what we should do is to get the integrated program, which covers the basic research and close to market innovation. That means that the research what we do in the university is kind of sometimes like really limited for a basic. But what European Union want at that time, that we do a research which should be marketed, like the common people can get benefit from this kind of research. So the total budget was 77 billion euros, which was still less than 10% of the public R&D spending in European Union. So the European Union decided to fund these projects based on three-pillar approach. What are the three pillars? I will discuss it in the next slide. So three pillars were excellence, industrial leadership, and social challenges. So they distributed the fund in these three different pillars. So excellence, this is kind of a basic research that is really essential to innovate. So ERC, which is European Research Council, so this is divided in these four different funding schemes. So ERC is for research fellows which are kind of starting now their independent research, like assistant professors also. So they get a starting grant of 1 million euros if they are able to successfully file this kind of application. Then there is a trend which is called future and emerging technology, which is kind of a basic research, but it's a little bit more in a technological field. So you get also funded for this kind of research. Then the Marie Curie Fellowship, which I got, it was also based on the basic research, but product driven, like which has some application. And some part of it is also divided in European research infrastructure. So you know that research is not possible if you don't have infrastructure, like if you don't have instrumentation. So European Union also gives fund for this kind of research. Then for industrial leadership, most of the fund goes to the key enabling technology. What are key enabling technologies? Key enabling technologies are like the technologies which are new and can be marketed easily. Then the risk and venture capital, which is like sometimes you open a startup company, and in the startup company you have more risk. But if succeeded, this kind of startup company can boost further and increase your GDP level. So in India also now we are getting a lot of fundings for this kind of ventures. So if someone wants to start their small companies, we are also funding quite well. Same thing goes in the innovation of KMUs. This is a key motive universities and units. Then the social challenges. So the fund is also going, like the highest fund goes on the social challenges. Social challenges means health issues. So to develop drugs for some diseases, food and security and safety. So also the research which is going in this field get funded quite well. Then the energy, what are the new resources for the energy, transport, climate change, society and security. So all these kind of fields get funded almost equally. So if you have a good research proposal, there is a high possibility that you get a fund to do the research and you get good infrastructure for that. Hope with that, I covered that which kind of research are kind of now going into the European Union in the different countries of European Union. And now I jump into the science. So science, I would talk about dynamic combinatorial chemistry and its use in drug discovery. So this is quite a recent technology. I will give you more details in the next slide. And then I will show how we were able to identify some compounds which stops the replication of coronavirus, which is called now SARS CoV-2, which is severe acute respiratory syndrome, coronavirus 2. So before going to that, I think everyone should know how drug discovery process work and what is the timeline required for the drug discovery. Many times you might wonder that why we don't have drug for COVID now. And you will get answered by the end of this slide why we can't get drug immediately for such a disease. So the drug discovery process starts by biological target selection. So drug discovery process is not a process for one field. It's a combination of different fields which involves biologists, computational chemists, organic chemists, pharmacists, doctors, people, government, and all the agencies which you can imagine. So before starting to any drug discovery project, there is a first thing which we have to see is if there is a target known. What is a target? Target is a protein, or we can call it enzyme, which is responsible for function of that pathogen. Pathogens are like bacteria or viruses or parasites, right? So this biological target, the task is covered by bioinformatics, genomics and genetic targets. So this is the field of biologists which bring forward this kind of target. So you remember that in 2020, in January, when the COVID started, At the end of January, there was a biological sequence of coronavirus was published. So it was published by a Chinese researcher, and then whole research field, whole researchers in whole world use these targets with this genome and identify different targets and search for therapeutics for this kind of target. So this is the task of a biologist. Then there is a second process, which is hit compounds generation. So what is hit? I will come back to this point several times in my talk. Hit and lead compounds. So hit compounds are not a drug-like compounds. They are really small compounds. So this is a starting point for the chemist. So you synthesize several compounds, you test them on a biological target. If this kind of compounds inhibit or stop the process, biological process of this protein. So there are different techniques which are used to generate this kind of molecule. High throughput screening. High throughput screening means generation of thousands of compounds in parallel. Then computational screening. So there are several computational chemist groups working where you don't have to synthesize compound, but what you can do is virtually use these compounds and see if they are binding to this target, right? So every target, like every protein, has some cavity where these compounds can fit. And then it goes to initial synthesis. So once you find that, okay, some of this compound which are virtually fit into the target, now we can synthesize them. And then assay development. What are assays? Assays are experiments. In a biological term, it's called assay. Assays are experiments where we check if the compounds which we synthesize, if they have a function, means if they can inhibit the function of a biological target, right? So high throughput screening at this stage, there are about 5,000 to 10,000 compounds synthesized, right? Synthesized or even purchased from some company, and they are tested in a biological target. Then out of this 5,000 to 10,000 compounds, there are lead compound generation. What is lead compound? Lead compound is like a drug-like compound. It's a bigger compound as compared to the hit compounds. And this compound has a definite activity towards the biological target. So here we started with about 10,000 compounds, and to getting to the lead compounds, we go around 250 or 200 compounds, right? And all of this compound are kind of showing their activity against the biological target. So at this stage, there is a requirement of advanced chemical synthesis where the organic chemist comes into the picture. And then there is structure activity relationship, which we look for. What is the structure activity relationship? Structure activity is like if you have a compound and it has different substitutions on ortho, para, or meta position, and we see that only ortho position compound is showing some activity on a biological target, and meta and para position compound are not showing any activity. So this is called structure activity relationship. So if you change the structure, activity change. So this we look into, that's the lead compound generation stage. Then there are drug candidates. So after going from 10,000 compound to 200 compound, it goes to the drug candidate. Drug candidates where you are definitely know that this compound will inhibit your biological target, but then biological target in most of the cases is in the bacteria or virus, right? Just consider, just for example, look into coronavirus now. So coronavirus, if you kill it outside the cell, like outside the human, it's possible. You can do it by the disinfectant, right? But you can't use disinfectant inside your body. So what coronavirus does, coronavirus enter inside your cells, and we have to get our drugs inside our cells to kill the coronavirus, right? So at this stage of drug discovery, where the drug candidates are found, actually we develop a cellular disease model. What is cellular disease model? Is like we use cells from, suppose, animal, like for instance, rat or mouse or chimpanzees or human. You take that cells, you put it in a petri dish, then you infect that cells with pathogens like bacteria or viruses, and then we treat them with drugs or the compounds which we synthesize. And at this stage, the drug should enter into the cell without killing the cell. So that is most important part. And it should kill inside the bacteria or the pathogen inside the cells. So this is called cellular disease model. So here we are not injecting the drugs directly into the animal, but we are taking the cells out of the animal, we are infecting them with the pathogens, and then we are treating them with the drug. And here we also see about the drug selectivity. Is this drug selective for this kind of disease, right? So suppose I have now one cell which is infected by several pathogens, like influenza virus, COVID virus, and then there are some respiratory viruses. We infect them, and we see that if these drugs are selective towards the coronavirus, right, for example, and this drug selectivity is measured. So the compounds which shows a better cellular disease model activity and a drug selectivity are then carried forward in this process. So going from 10,000 compound to 200 compound, these drug candidates are around 4 or 5 compounds, maximum 5, not more than 5 compounds. Then there comes the preclinical development. Preclinical development, as I said before, it was a cellular model. Now this is an animal model. So what we do, for instance, in the rat or mouse, we infect them with, for instance, for example, just consider now the corona situation. We infect them with the corona. And then we try to use these 5 drug candidates and give them orally, intravenously, or intramuscularly, right? There are different pathways where we can administer this drug to this animal model. And then we see if the coronavirus inside the human or inside the animal is killed or not. And there we go for pharmacokinetic and pharmacodynamic studies. So for the bachelor and master student, I would like you to look into these two terminologies, pharmacokinetics and pharmacodynamic. So what are they? So pharmacokinetic is kind of how your body reacts to the drug, like how it metabolize your drug. Pharmacodynamic is also like what is it if you have a concentration effect of your drug. So if lower the concentration, lower the effect, higher the concentration, higher the effect inside the body. But to know it in more details, you should read it carefully. Then we also check if there is a toxicity of these compounds to the mouse model, suppose. So if we incubate our mouse, or if we inject this mouse with the drug candidates, and if it dies, then this is not a good drug. So we have to get it back, right? So then we also check a behavioral study, like what behavior these animals do. Like after giving this drug, if this animal behave really weirdly, so that is a no go. So if the animal behaves normally, then okay, we will carry forward the drug. If not, we will stop this drug and then go back to the synthesis again to synthesize different compounds and then go back. So this is kind of a cycle. So we do chemical synthesis, we get their structure activity relationship, and most of the time spent in this period is years. I will show you it in my next slide, that how many years required to go from the hit compound to the preclinical development. After you get a compound which shows really good activity in the animal, nice pharmacokinetic, pharmacodynamic, and the animal model is not even like animals are not behaving weirdly, then we carry forward these drugs in the clinical trials. So clinical trials, these are the human clinical trials. So there are 3 phases of it. Usually till here, we can do everything in academia, right? But to do the clinical trials, we required a huge fund, like billions of dollars, billions of euros. And here we have to somehow get industry into the action. So that's why we see like a lot of pharmaceutical industries, they are doing this kind of clinical trials. So these clinical trials are divided in phase 1 to 3, which are depending upon the number of people or number of patients which you put into the clinical trials. The phase 1, there are less people as compared to the phase 3. So when this already proven drug in the animal, we will put them into the human, so infected humans. For instance, if you have now corona, patients from corona, we put this drug into them and then see if they cure it. So this clinical trial also requires years. Then once this clinical stage is passed, that there are no adverse effect on the humans which were treated, then it goes to the government approval, government agency. So in US, you have FDA, Food and Drug Administration. In India, we have different agencies which do this kind of drug approval. So we have to go to them and we have to tell them, okay, we have this kind of study, and then it's called ICMR, yeah. So ICMR will then check into your study data and see if this drug is approved or not. And when it's approved, then it goes into the market. So after market, the whole drug discovery process is not end yet. So then there is a post-market safety surveillance. So which is like, you put this drug in the market, you can buy it from the pharmacy, and then you check like real-time uses of this. So suppose if the drug is now in the market and then many people are using it and now finding like most of them are getting sick or most of them are having some side effects, then many times this marketed drug is withdrawn from the market. There are several drugs, several examples of this, that the people or the company spent like 10 years in developing this kind of drug, but after even marketing, we found that they are not good enough. So they have to brought them back from the market. Then I hope that I covered how the drug discovery process works. Now we should look into what is the timeline required for this kind of research. So the pre-discovery stage, which is like a biological target selection, can be done within months. That's not a problem. But then it comes to the drug discovery and preclinical trials, which requires around 3 to 6 years. So drug discovery, which contains hit, compound identification, lead generation, and drug candidates, right? These 3 steps, and then the preclinical trials in the animals, not humans, it requires 3 to 6 days. And as I told before, we start with around 5,000 to 10,000 compounds, like these are the hit compounds, then go to the lead compounds in preclinical trials, which are around 250. Then we go to phase 1, which is human trials. These are like 4 or 5 drugs candidate, maximum 5 drugs, which we can get. And then in the whole 3 phases of the clinical trials in human, it also requires 6 to 7 years. And after that, it goes to the review, to the government industry, and then large-scale synthesis and marketing. And here, only 1 drug can be approved. So starting from 10,000 compounds to 1 drug, which requires average of 10 years of research. That's why the question, whenever someone asks you why there is no drug for COVID, because COVID started now, 2 years before, there are companies which are working for it. Now, this period is now kind of reduced. So we will require maybe at least 5 to 6 years to get this kind of drugs in the market. So I hope that this covers how the drug discovery process works. And for many of you, this is kind of information which you never received before. But let's go to my institute, what we do in my institute. So I'm working, as I told you, in the Department of Drug Development and Optimization. So we focus on this part of the drug discovery process. So we have biologists in our floor, which selects the target. Then we have chemists which synthesize the compounds or check for the compounds and go from the hit compounds to the lead compounds to the drug candidate. For preclinical trials, they are not done at this institute, but we have a sister institute which are focused on this kind of preclinical trials. That's why I put it this box, half of the preclinical trials. But that is also in the academic setup. And this is like this first 3 to 6 years or 5 years, we do it in our institute. And what I am focused on is to identify these hit compounds and generate the lead compounds. So my goal is to find alternative strategies, so alternative ways to find these hit compounds. and then optimize them to the lead. And the goal is to reduce this 3 to 6 year time to 1 or 2 years. So that is the most important part for faster drug development. And there I usually, my research lies on this area. So I told you about the hit compounds. So from where they come, like usually they come high throughput screening, like thousands of compounds are screened, or by the computational screening. But from where the basic idea comes from, like from where the structure, suppose if you have the structure of compound, from where it comes. So you will be surprised that most of it is come from the known compounds, so known drugs, right? So around 43% of this hit compound come from the known compound which shows some activity in a previous research. Then it goes to random high throughput screening, as I show here. So thousands of compounds are tested against the target, which is the protein. And then there are some direct approaches. I will not go in details about them, but the common function of all these different approaches, they are traditional ways. All the traditional ways, and they required more time to generate. And most of the time, the quality of these hit compounds are not that good. And then you have to go backward and forward into the drug discovery process to find the best compound. So as I told you, here I introduce my research, like how I reduce this time. So instead of going to this whole traditional ways, I will show you a new way which can generate a high quality hit compound. What is a high quality hit compound? High quality hit compound is the compound which shows a really nice activity, but it's on a target. And it has more chances to go through whole clinical phase, so whole process without failing. And here it comes, the target directed dynamic combinatorial chemistry. So what is the unique of this chemistry? Here the biological target, I will discuss in details about what are the biological targets in my next slide. But just consider biological target is a protein inside a bacteria or virus which is responsible for some of their function, right? For replication of their function or something like that. And here in this technique, we use this biological target. We throw some ligands inside that. And each biological target has some cavities where these compounds can bind, right? So what happened that this biological target, when we put some compounds, it kind of brings them together and the reaction occurs. So this biological target can also act as a catalyst for your reaction because it's bringing two of the reactants together. And that's how it's forcing them to react. And when it reacts, it forms a new molecule. That molecule is fitting into the cavity of this biological target. And if it fits into the cavity of biological target, most probably that it will also inhibit, like stop the function of that biological target, right? So for example, so we use reversible reactions. So reversible reactions, this is acyl hydrazone formation reaction, which starts from aldehyde and hydrazide. So when they react it in an acidic pH, this reaction is reversible. So you will get always the reversible reaction when it is in an acidic condition. So for simplicity, how this approach works, what we do is, so this head of these keys, right? These are the keys. So head of these keys, just consider these are aldehydes. And this tail of these keys are hydrazides, which are depicted in blue. So in one of the, in one vial, we throw all of them together. And when we throw all of them together, it tends to form different combination, right? So we have 2 aldehyde and 4 hydrazides, so they can form 8 different products, right? And that 8 different products and these reactants, they are always in equilibrium. And this is called thermodynamic equilibrium. So what we do, so whenever we throw external stimuli like a protein, right? Protein from, or the biological target, that's what I'm mentioning, protein into this library, this equilibrium shift towards right. How it shifts towards right? Because what is happening now? So suppose this second key head is fitting here, and then the second key point is fitting here. So what this protein is doing, so instead of choosing any other things, it's choosing these two components. They are bringing it together, and it's forcing them to form faster than the other products, right? So here is a nice example. So this is a protein target, and you can see there is, in the pink, there is this molecule. So this molecule fits into this cavity, and this cavity is responsible for one biological function of this target. This target was from a bacteria. So what happens now? Yeah. So as I told you that now this biological target force this compound to form over all the other compounds. And then what happened, that you quench this reaction. How to quench reaction is to kill this protein, which is called protein denaturation. We kill this protein, we raise the pH to 10, like basic pH. In basic pH, this reaction is no more reversible. So whatever you form here, it's frozen now. So when it's frozen, that means that these compounds are final compounds, and they will not go back to the aldehyde and hydrazine. And here, then you analyze this library. And in the library, you see that this compound is highly amplified or highly prepared over the other, right? It's 30%, and all the others are 10%. So then we analyze it by HPLC-MS, so high pressure liquid chromatography, which is coupled with mass chromatography, right? Mass spectrum. So there we identify, okay, this compound looks like it's amplified a lot. So it's produced more than the other compound. That means that this compound is now fitting well to the target. So how do we perform this kind of reactions? They are really simple. They are micro scale reactions. So you work on around 100 micromolar concentration of this compound. So we set two reactions. So one is blank, where only, suppose, aldehydes and hydrazides, only the fragments are put into this kind of Eppendorf, which is called Eppendorf, this plastic tube, right? And in the other, we put these fragments plus protein, which is your target. And then when we analyze after the reaction, after the protein denaturation, and then basifying the library, so freezing the equilibrium, we check and we compare the blank reaction with the protein templated one. And when we check the blank reaction with the protein templated, we see now these peaks belongs to products, right? So you can see number 1, number 2, and number 3, these peaks are higher as compared to the blank library. So what does it mean? This means that these 3 compounds were catalyzed by the protein. In other words, these 3 compounds are fitting well into this pocket or the cavity of this protein. And then instead of synthesizing now these 8 compounds, like in a large scale, we just synthesize these 3 compounds. And that reduce the synthetic efforts. So for instance, in a traditional way, what you will do, you will synthesize all 8 compounds, you test their biological activity, and then found out, okay, maybe none of them are active, right? In this case, you will synthesize only the compounds which are catalyzed by the protein, the target itself. And most of the time, these are the compounds which are really active. So what it does, it reduces the synthetic efforts in the process of finding drug candidates. So less synthetic efforts, better quality hit you are getting. And most probably that this hit will not fail in the drug discovery process. And at the final stage, it will shorten the period, which I showed, like this 3 to 6 years, right? It shorten this period of iterative cycle of design, synthesis, test, and analysis. This will reduce. So I hope that now I cover how this kind of technique is used to find the hit compounds or the good quality hit compounds. Hit compounds are the compounds which are used at the beginning of the drug discovery process. Now let's look at the example, right? So for example, I will show you, we use SARS CO2, that is a coronavirus. And I think most of you know what is coronavirus by now. So coronavirus, now I was showing you, talking about biological targets and proteins, right? And I will show you now what are they, where they use. So with the example of coronavirus. So coronavirus has 4 structural proteins. What is structural proteins? They are responsible for its structure, the assembly of the virus. So first one is the spike protein. Most of you kind of heard about the spike protein on the coronavirus, right? This protein is responsible for formation of this kind of spikes on the coronavirus. And these spikes are responsible to enter into the host cell, like in our case, like human cells, right? Without this spike proteins, this virus can't be enter into the human cell. Then there are 2 proteins which are called envelope protein and membrane protein. So envelope and membrane protein are these 2 proteins which are responsible to form this spherical shape of this virus. So without this protein, this spherical shape will not form. So this is the function of this 2 proteins. And then there is a 3rd structural protein, which is nucleocuspid protein, which covers the whole RNA strand, right? The coronavirus is a plus sense RNA virus. Plus sense RNA virus means its RNA is coiled at the clockwise direction, and it has only one RNA. So this coiling and the structure of RNA is kind of governed by this nucleocuspid protein. So you know that when everyone says like, oh, wash your hands with soap to kill the coronavirus, what it exactly does? It exactly does this membrane and envelope protein. They are lipophilic proteins. And when we use soap, they kind of break this lipophilic protein. And then by breaking this lipophilic protein, this coronavirus is bursted and open, and that's why it's killed and it's not able to replicate further. So we talk about 4 structural proteins, right? And most of the time, you know, like all these vaccines which are now generated, they are for the spike proteins. So they are using this spike protein as a vaccine vector. So after this structural proteins, I will show you what are other non-structural proteins. So non-structural, in coronaviruses, there are 16 non-structural proteins, which are called NSP, non-structural proteins. Each of these proteins have a different function. And the most important part, that all these proteins are only expressed inside the host cell. So these proteins are not expressed when the virus is on like a surface or in the air. No, they are only expressed when the virus is inside the cell. What are their functions? So most of the time, the function of all these non-structural proteins is to transcript. Transcript means cut the RNA into fragments, and replication. Replication means to find these fragments and generate a new RNA virus, right? So all these non-structural proteins are responsible for transcription and replication of RNAs. And then when these transcripted viruses or transcripted RNA are like form a new RNA, then these non-structural proteins, these 4 structural proteins play their role. So they form this assembly. So this RNA is encoded now. So this RNA was formed by the non-structural proteins, and the structural proteins now encapsulate it, like cover it, and form this whole envelope, right? And this is how the replication of coronavirus is done. So I will not go into each of these non-structural protein and what their functions are, but I will show you some of them, like 2 of them. As I already told you, these non-structural proteins are expressed inside the host cells and very important for viral replication. So just remember it, the non-structural proteins are responsible for replications of virus, and the structural proteins are responsible either to enter into the host cell or the assembly of the coronavirus, right? And one of the most important point which I want to highlight here is that all these non-structural proteins are highly conserved. What is highly conserved means? So highly conserved throughout the different coronaviruses. Most of you might know that we have 4 different coronaviruses which are present around us and which are known as like common cold viruses, right? But they are not that pathogenic. They are not that dangerous like SARS-CoV-2. So there are these 4 coronaviruses, the old coronavirus 1 and this new coronavirus. In all these coronaviruses, these non-structural proteins are highly conserved, means they are not changing. So they have more than 90% of similarity. So there is less mutation going on or no mutation going on for this kind of non-structural proteins. On the other hand, if you look at the spike protein, you know now the Delta virus, which is revolving around the world, is now due to the mutation on the spike proteins. So if you try to develop a drug for a spike protein, most probably it will not work after a few years or even after a few months. So there are a lot of research groups which are also working on this kind of field, like developing a drug for the spike protein. But why we choose non-structural proteins? Because if we prepare a drug for a non-structural protein to inhibit its function, like if I now talking about the preparation of drug, that means I'm talking about preparation of chemicals which can inhibit, which can stop their function, which is transcription, like breaking of RNA, and replication of RNA. So if we kind of stop this function, we might have chance to develop a broad spectrum antiviral drug. What is broad spectrum antiviral drug means? That means that it can be effective not only for the SARS-CoV-2, that is the coronavirus 2, but it also responsible for the old coronaviruses or the common cold viruses. Most of the people knows that there is no cure for, or there is no drug for common cold, right? So this kind of a drug discovery approach and use of this kind of non-structural protein is really important. And I will talk about non-structural protein 14, number 14 and number 10. So this is the protein, and I will talk in details about what is their function. And now you will know what are the biological targets and proteins I'm talking about, right? So this is how the protein looks like. So in the green, this is Nsp12. So Nsp is non-structural protein, Nsp12. And in the red, there is Nsp10. So this is a complex of Nsp10 and 14. So what is the function of Nsp14? So Nsp14 has 2 activities. So it has the first activity, which is N-terminal 3', 5' exoribonuclease activity. What does it mean? That to simplify its terms, like both these activities are the activities which are responsible for transcription and replication of virus, like to cut the virus into the pieces and to assemble this virus into a new RNA virus, right? Like just for the RNase. So these activities are for transcription and replication of RNase. And Nsp14 is responsible for this kind of activity. But what is the role of Nsp10 then, if the Nsp14 is having this activity? Actually, Nsp10 acts as a ligand for this Nsp14. So without Nsp10, there is no activity of Nsp14, right? So they should be into the complex to get this activity done. And there was a recent study in 2020 which shows that they prepare coronavirus in lab, and they use the knockout mutant. Knockout mutants mean they perform a mutation that this coronavirus is lacking the Nsp14 protein, right? And this coronavirus, which were lacking with the Nsp14 protein, were not viable. Not viable means they are not able to replicate. They are not able to leave into the host cells. So that means that really Nsp14, that shows, or that is the proof that Nsp14 is really important for the virus assembly and replication and its survival. Then there is a recent study which is published in 2021, which shows that this activity, this Nsp14, is not only responsible for the RNA cleavage and RNA formation, but it also responsible for reducing the host immune system, like reducing the host immune response. So due to this protein, when it is inside your body, coronavirus, and when this protein is expressed inside your body, it also kind of trick kind of change your immune system in a way that your immune system will not react against this coronavirus. So these are the different function of this Nsp14 protein, and if we stop this function, most probably that the coronavirus will not be able to replicate. So then it comes to which of this Nsp14 and Nsp10 we select for the drug discovery projects, right? So most importantly, I told you that this kind of proteins can be generated by biologists, like they can be expressed, purified by the biologist. And most of the time, these proteins are not stable at room temperature. So we have to work really at a lower temperature and everything. So in this case, one of our colleagues in our institute is able to purify both of this protein, Nsp14 and Nsp10. Then the problem was Nsp14 was not that stable, but Nsp10 was. So we decided to use it. And there is another reason why we use it. So Nsp10 is a small protein as compared to Nsp14, which is really big, right? And the active site, or I can say the cavities, right? Cavities in this protein, which you can see here. There your chemical compound will bind. So these cavities are located only on the junction of this ligand and the protein, right? This protein-protein interaction site. So most of these cavities in Nsp10 are only responsible for formation of this complex. On the other hand, if we consider the whole Nsp14 is a very big protein and it has several of this kind of cavities. And if we use that protein for the drug development project, most probably what will happen that your compound will bind to every active site or every cavity, right? But this is not the case for Nsp10. So if we bind our compound to this cavity, it will not bind to the Nsp14 complex. So this complex formation will not happen. So if you put your compound here, right? And the idea then is to prepare the drugs for Nsp14, the compounds which binds to Nsp10, sorry, and then the complex formation will not occur, and then the whole viral replication and transcription will not happen. So that's why we choose Nsp10 over Nsp14 for our experiments. So first thing is to check all the biological targets, all the biological proteins are most probably not compatible with your organic solvents, right? Fortunately, this acyl hydrazone formation reaction is really good that it can be performed in water with a really slight amount of DMSO, which is 5%, right? So what we check, that we have a random library. So we use 3 aldehydes. Unfortunately, I can't show you the exact structure because of the privacy law at my institute. But just to give you an idea that we use 3 aldehydes and 7 hydrazides, right? And then we add Nsp10, which is our protein. And the solvents where PBS is the phosphate buffer, which is in the water, DMSO, and aniline was the catalyst for this reaction. Because at higher pH, this reaction is not reversible, right? That's what I told you. But when you add aniline, which converts this aldehyde to imine, and then imines are more reactive than the aldehydes towards this acyl hydrazone formation. So, and in that case, the reaction is still reversible. So that's the role of aniline here. And what we found out that for 48 hours in this reaction condition with this different reagents, and this protein Nsp10 was stable. So it was not degraded, right? It was not decomposed. And we also saw there's amplification. Amplification means like amplification of some of the products. So in total, you will get from 7 aldehyde and 7 hydrazide, 21 acyl hydrazones, right? So out of this 21 acyl hydrazones, there were several acyl hydrazones which were amplified over the others. And I will show you in the next slide how they are amplified. So this is the LC-MS chromatogram, right? Yeah. So LC-MS chromatogram. So this is a blank reaction. Blank reaction, I told you how we perform the blank reaction, where only aldehyde and hydrazides were used. And this is a protein templated reaction where aldehyde, hydrazides, and a protein is used. Then these reactions are run in parallel. They have exactly the same concentration of each reactant. And then after some time, we quench the reaction. We quench the reaction means we denature. You see that this peak at 12 minutes is higher than the peak in the blank reaction. Same goes for the other peaks, other green peaks. So this means that this compound is amplified, or it's catalyzed more by the protein. So on the other words, these compounds are binding to the active site of the protein, right? And to simplify this, I prepared this bar chart, and where higher the bar chart, higher the amplification, right? For instance, with this first compound, which is at 1.5 minutes, which was not even present or really slightly present in the blank reaction. But when we add a protein, it increases a lot as compared to the other compound. So that means that it's highly amplified compound. That means that it's strongly binding to the protein. And same goes for all the green, these 4 compounds, which were highlighted in green. They were amplified more than the other compounds. And then there are some 3 compounds which are amplified a little bit. They are amplified, their peak area is a little bit higher than the peak area of the compounds in the blank reaction. So I'm sorry that I can't show you the exact structure again, but just to let you know that these were the 4 compounds which we synthesized them, because instead of synthesizing all these 21 compounds, we just synthesize the compounds which are amplified, right? And these are now in the real chemistry reactions, like large-scale reactions. We work on a gram scale before we work on a microgram scales, right? So we synthesize only this compound. In this way, we also reduce the amount of reagents we require. So we have less money invested in this project as compared to the traditional medicinal chemistry project. And most probably whatever hits which we are generating from here, or hit compounds which we are generating here, they will be active because they were selected by the protein target, right? So how do we see that if these compounds are really binding, right? There is a surface plasmon resonance study which we use to know the kinetic. So it is KD. KD is called, there is dissociation coefficient. So lower the dissociation coefficient, better they are binding. So all these 6 compounds shows they were binding at a different level. So this compound, number 1, binds better as compared to the other. Then we also use if they are binding by the MS, so to form the complex of Nsp10 and this product, and check by the MS if we observe the mass of this complex. And we found most of them were binding to the Nsp10. Then what happened to the final coronavirus? So we have a collaboration with Professor Thomas Pichman from TwinCore. So TwinCore, where we have this virus lab, so viral lab, which is a safety level 3 lab. So where what they do, they use the cells from the animal, they infect it by coronavirus, right? And in this case, it was the old coronavirus, the human coronavirus 229E, which was easy to handle as compared to the new coronavirus. So they incubate, they infect these cells, and then they add our compounds to that, and then see if this coronavirus is killed. So these 2 graphs, which you can see, the lower the graph, the green one, which shows the infectivity of the coronavirus, which is reducing now. So this is a concentration of your compound. So once you increase the concentration of compound, the infectivity, the virus load is going down. And the other one is the cell viability. Like, you don't want to prepare any compound which kills your cells, right? So it's higher this value, better it is, and lower this value, better it is. So this shows that at least 2 of these compounds which we synthesized were able to kill the coronavirus in the cell. So, and now we are going into the Sars Co2 assay. So this is the new assay now, which is developed in this institute, and we will test all our compounds on a real coronavirus now. So this brings me to the conclusions and outlook. So I hope that I show you that the target-directed dynamic combinatorial chemistry is applied well for this NSP10 target from the coronavirus 2. We also found the 4 binders for this protein, and using this approach, and 3 of them shows in vivo activity, so in cellular activity against this old coronavirus without any toxicity. And now we are also looking into the actual activity of this compound, and this compound will be tested on the new coronavirus, and then further optimization will carry out. So this is the end of my talk, but before going to the acknowledgement, I would like to show which are the opportunities for the young and enthusiastic researchers abroad if they want to pursue PhD or MS masters in abroad. So there are a lot of funding agencies which you can apply for. Yeah. So I will talk about each country. So for Germany, there is a DAD fellowship, which is a fellowship, and you can take a screenshot afterwards for this slide when I finish this slide, and you can go to the particular website and to look into the details. So that fellowship is for the master's and PhD students. So who has the capability to do the independent research, they can apply also for this DAT fellowship, which is basically from Germany. Then there is a Denmark government scholarship. Then there is also a France Eiffel scholarship, which is called, and it is also for the MSc and PhD. And this Denmark scholarship is also for bachelor's, master's, ME, and PhD. Then Holland is the Netherlands, where I also worked. So they also have a nice set of fundings for new researchers. Then there are also in Italy, for bachelor, master's, and PhD, you can look for this Italian government site. I will give you the site at the end of this slide. In Sweden, in Switzerland, and in Belgium, these are fully funded scholarships. They are really highly competitive. You need to be really scholar to get this kind of scholarship, which are bachelor, master's, and PhD. And then in UK, United Kingdom, there are this Bill Gates scholarship for the Cambridge and Rhodes Trust scholarship for the Oxford. And all this information you can find on this site. You can note it down. But before going to that, I would also like to tell you what they are looking for, right? Because not everyone can get this kind of fellowships. So what are the requirements? So the most important requirement, they need someone who has knowledge of research project. So who have high quality research project in MSc. So preferably from the well-known research institute or industry. So I would encourage for the master student, if it is possible, then try to do, some of you try to do your project, master's project into the industry or the institutes like National Chemical Laboratory, IICT Hyderabad, or there are some pharmaceutical companies where we are also promoting this kind of research project. So please go there and do the research, and then you get exposure to different instruments like in experience. So they also want people who has a basic knowledge of analytical instrument like GC, LCMS, NMR, this kind of thing. And you will get this kind of exposure in this kind of institutes, right? Then you should have a basic knowledge of literature search, how to do the literature search. That is important because they will check it during your interviews. They will check how do you write your proposal. Then publications are not required. Like you shouldn't have publications, but if you have them, like publication in international peer-reviewed journals, not into the conferences, right? So if you have publications in international journal, they are highly possible that you will get a position. Then you should have a good future research project. So you have to write a future research project to get this kind of grant application. So you write a nice research project, they will evaluate, of course, your academic record. if you have a nice research project done before, if you have experience, and if you fit into all these kind of requirements, most probably there is a chance that you can get one of this kind of fellowship. So go through this site, and maybe you can find, if you have some problems, you can also contact me. I can guide you through how to do this process. Okay, this brings me to the final slide, which is acknowledgement. And I would like to acknowledge the people from my department, like from Professor Anna Hirsch, who is the head of the department. Then Dr. Sebastian, who is doing this protein purification, whatever proteins I showed you. Then Sandra, she's a PhD student who is working on finding if these compounds are binding to the protein. Then at TwinCore, as I told you, they have a safety level 3 lab where these coronavirus assays or coronavirus experiments are running. And this is with the Thomas Pichman lab. And there are 3 students who are working on that. Then for another MS, neonatal MS, we have a group on the 3rd floor of Professor Rolf Muller and 2 of his co-workers. And they were also highly acknowledged for their help in this project. And fundings, yes, Helmholtz Association and Saarland University for funding this research. To fund my research, Marie Curie, I highly acknowledge the funds which European Union gave me, and the European Research Council for funding all these research projects. Then I would like to also thank the organization committee and Dr. Vivek Shinde, President of BHADRAWATI Section Society, Dr. Kartik Shinde, Dr. Vishal Shinde, who are Joint Secretary and Joint Secretary respectively, and Professor L. Larkeza, the Principal of NILKANTHRAO Shinde Science and Art College, for giving me this opportunity. Of course, the chief guest, Professor Dr. Thairesa, organizer, Professor Dr. Larkeza, and whole organization committee, Dr. Aparna Madam, Dr. Gajendra Sir, and Dr. Kiran Madam, for organizing this webinar and giving me opportunity to work. And of course, all the virtual attendees for